Hey everyone, welcome back. In previous videos, we have discussed in quite a lot of detail the process of not only cell division, both mitosis and meiosis, but also Mendel's work in establishing the laws of inheritance. So in this video here, we're going to continue on with that. A lot of our previous discussions have been basically assuming that everything is going to go correctly, chromosomes are going to segregate properly, and everything is going to be hunky-dory. But in this video, we're going to talk about, well, what happens when chromosome segregation, mitosis, and meiosis don't exactly go correctly, especially when we talk about meiosis. So you'll recall we've talked about a karyotype before, which is basically just kind of an organized photograph of all the different types of chromosomes that you find in a cell, and we do this for a variety of reasons. It's not just to get a pretty picture. We do this as a way of identifying some really important genetic parameters, such as how many different types of chromosomes are there? Do we have two copies of each of them? How long are the chromosomes? Have we lost any pieces of chromosomes, like we'll talk about later? Are the centromeres in the right places? What do the banding patterns look like? So if you're talking about a human, we've said before, humans have a diploid number of 46. So a haploid number of 23 means that if we do a karyotype on a human, then there ought to be 23 different types of chromosomes, and there ought to be two copies of each one. And you can see that that is the case for this karyotype right here. Now, in terms of how to actually perform a karyotype, number one, this requires the isolation of DNA from cultured cells. So you need to actually obtain cells from a living individual that you want to karyotype. Usually the best way to do this is to get a blood sample and then to use some white blood cells. You can't use red blood cells because red blood cells do not have nuclei and therefore they do not have DNA. So the question here becomes, and this is kind of an important review question from a previous chapter, in order for the chromosomes to be visible like they are in the picture above, the cells have to be at which stage of the cell cycle? So another way of asking this question is, could we just obtain a whole bunch of white blood cells from someone, and could we bust them up, isolate the DNA, and expect the chromosomes to look exactly like they do here? And the answer is no, they cannot. So you'll recall that condensed chromosomes are only visible during mitosis. So in order to actually get a picture like this from DNA isolated from living cells, we have to find a way to get all of those cells synchronized in mitosis. So a way that you can actually do this is to treat these cultured cells with a drug called colchicine, or there's another drug that does the exact same thing called nicotazole. So what these two drugs have in common with each other is that they are both disruptors of the microtubule machinery. Microtubules, intact microtubules, are built by polymerizing small little proteins called tubulins. So if you want a long, stable microtubule, you need to get a bunch of those tubulin proteins polymerized together. What these drugs do is they actively depolymerize the microtubules and they prevent microtubules from forming long, strong, stable fibers. So, because you're treating cells with drugs that disrupt the microtubule machinery, we cannot get the chromosomes lined up at the metaphase plate. You'll recall from chapter 10 and chapter 11 that the microtubules latch on to the kinetochores, and those pulling forces on either sides of the sister chromatid pairs, those are responsible for lining the chromosomes up at the metaphase plate. So if the microtubules are disrupted, we cannot do that. And because of that, all of the cells are eventually going to all get arrested at the spindle checkpoint. Now, if you have a collection of, let's say, several trillion cells, and, well, maybe that's too many, maybe several million cells, and, you're asking, well, maybe some of them are in mitosis. Well, yes, you are correct. I would bet out of a million cells, I would bet probably at least several thousand are in mitosis, but you want all of them to be in mitosis. So maybe a few thousand of them will be in mitosis. The other several 100,000 of them are going to be in interphase. So when we say synchronization here, we are saying we are going to treat all of the cells with the same drug that are going to disrupt the microtubules, So the cells that are just about to enter mitosis, they are going to get arrested in metaphase first. And then as all the other cells go through interphase normally, once they get to mitosis, then they're going to get arrested too. So if we treat with this drug and we wait several hours, eventually all of the cells are going to be synchronized and they're all going to be basically on the same schedule in terms of where they are in the cell cycle. They're all stuck at the same point. So the whole idea is that since this spindle checkpoint arrest happens after prophase 1, which is when all of the chromosomes condense into visible structures, all of the cultured cells will now have visible chromosomes that we can then bust the cell open, disrupt the plasma membrane, pull those chromosomes out, lay them down on a substrate, and then take a picture of them and stain them. And then we can use like something like Photoshop to get an actual picture where they're all lined up in a nice, neat row like you see it right there. Okay, so talking about something a little bit new, and this actually does have something to do with karyotyping. Back in chapter 11, we meticulously went through all the different stages and steps of meiosis, and what we can kind of understand about that is, under most circumstances, meiosis almost always proceeds normally without any sort of problems. So when that occurs, when everything happens normally, we use the term euploid or euploidy to refer to cells that have the correct number of chromosomes for that species. So a somatic cell should have 46 chromosomes if that cell came from humans, and then a human sperm or egg should have 23 chromosomes. If they have the correct number of chromosomes, those are euploid cells, and it indicates that all the different stages of meiosis went correctly. Now, a different term that we sometimes use called aneuploidy, this is instead used to refer to cells that for whatever reason have the incorrect number of chromosomes, whether it's too many or too few. This is usually because of some sort of problem that occurs during meiosis. And the reason why we wanted to talk about karyotyping first is because a karyotype is actually a very good and reliable way to confirm this. If a cell is aneuploid in nature, you ought to be able to look at all the chromosomes and see that you've got one too many of one chromosome or maybe one too few of another. So the cause of aneuploidy is called nondisjunction. This is a failure of chromosomes to separate properly during either meiosis 1 or meiosis 2, and it can occur in either case. So the result here is going to be that you're going to have daughter cells that either have too many or too few chromosomes rather than the correct number. So in most cases, you can end up with one of two different problems. In the case of monosomy, a cell will only have one of a particular kind of chromosome, or in trisomy, the cell can have 3 of a particular kind of chromosome. Now, given previous discussions we've had about gene dosing, we've talked about things like haploinsufficiency, the whole idea that if you have a recessive allele that is non-functional, then you're not getting any functional protein made off of that particular chromosome. So you're basically dealing with 50% protein dosage. So based on that discussion, you can kind of imagine how monosomy would be a big problem. Basically, for every gene that is on that chromosome, you are only getting 50% of the normal expression. So it's kind of not hard to picture why that would be a big problem for a cell, only having 50% of the normal gene dosage. Now, what can be a little harder to picture is in the case of trisomy. If you have 3 copies of a particular chromosome rather than 2, you've basically got 150% gene dosage. So you might be thinking, well, that's great. We got more than enough protein to go around. Well, I'm not going to get into specifics because the implications are a little bit different for every gene and every protein, but having too much protein can often be as big a problem as having not enough. So neither one of these situations is very ideal, and you would much rather have a situation where you only have 2 copies of each chromosome rather than 1 or 3. This diagram here actually shows you the different ways in which nondisjunction can occur. So on the left, you can see a graph of meiosis in which the nondisjunction event occurs during meiosis 1, in which we don't do a good job of separating homologous pairs from each other. So you can see that the entire homologous pair goes to one of the daughter cells, but not the other. So that by the time we get through meiosis 2 and we have a fertilization event take place, we have 2 of our cells have a case of trisomy, and then 2 of the other daughter cells have a case of monosomy. It is very, very, very bad to have a non-disjunction event occur during meiosis 1 because you are pretty much guaranteeing that one of those daughter cells after fertilization occurs is going to be aneuploid in nature, whether it's trisomy or monosomy. It just depends on which one of those eggs the sperm fertilizes. Now, in the case on the right, you can see that meiosis 1 actually does go off without a hitch, and we do a good job of separating homologous chromosomes from each other. But once we get to meiosis 2, in one of the cases, we have a non-disjunction event happening that causes us to not separate sister chromatids properly, so that from this daughter cell, we have one cell that after fertilization is going to be trisomy, and then the other one is going to have a case of monosomy, but the other set of daughter cells are actually normal, because presumably, based on the rarity of non-disjunction, you would assume that non-disjunction would only happen in one of the daughter cells in meiosis 2 rather than both of them. So if you had to choose, you would much, much, much rather non-disjunction occur during meiosis 2, because there's still, in this particular case, a 50-50 chance that after fertilization occurs, your offspring, your zygote, is still going to be euploid and not aneuploid. Okay, so as we said before, trisomy occurs when an individual has 3 of a particular kind of chromosome. Now, in a lot of cases, monosomy is a big, big, big problem for what we said before, because you have only 50% as much gene dosage as you normally have. So in trisomy cases, in most cases, autosomal trisomies are viable beyond birth, meaning that if you do have an offspring that has a case of trisomy, they're probably going to survive post-birth. The most common autosomal trisomy, you've probably heard of this before, is trisomy 21, and it's also called Down syndrome. So I'll show you a graph here in just a minute, but oftentimes what we see is that the probability of a woman having a child with Down syndrome tends to increase with age, meaning that the chance of a 20-year-old woman having a child with Down syndrome is significantly less than a 35-year-old woman having a child as well. So the idea here, if you remember how oogenesis works based on what we talked about in chapter 11, when oogenesis occurs, there are a lot of pauses, meaning that there is a pause in meiosis 1 while the female in question, it has not been born yet. And then we hit the play button again to continue on through meiosis 1 once that female goes through puberty. And then after that, the developing oocytes will then pause in meiosis 2 until fertilization occurs. So basically you have a lot of produced oocytes being stored up, and the idea here is that the longer they get stored up, the greater the chance of non-disjunction occurring. So we can recognize Down syndrome in particular based on a number of different physical characteristics, not even talking about being able to identify it with karyotyping and DNA analysis, but oftentimes those affected with Down syndrome will show some physical characteristics like short stature, a folding of the eyelids, kind of a flat morphology of their face, kind of short stubby fingers, and then kind of some morphologies of their feet and their toes. So what we can kind of assume here is that the problems associated with either trisomy or monosomy are related to gene dosage. So like I said, with trisomy, you've got 50% too much protein, and with monosomy, you've got 50% too little. So for Down syndrome in particular, you can identify it with this karyotype right here. You can see that this person has the correct number of chromosomes for every chromosome except chromosome number 21. So this male here, we can tell it's a male because of the X and the Y chromosome. This male has an extra copy of chromosome 21, so that should be enough evidence to go ahead and make a diagnosis of Down syndrome. Now here is that graph that I mentioned before. You can see that in the early to mid-20s, the likelihood of having a child with Down syndrome is very, very minuscule, but that chance goes up exponentially the longer a woman waits to have children. So you can see when we wait until, like, say 45 years old, which in that case, being able to have children is kind of a rare event in and of itself, but the risk associated with having non-disjunction events increases quite a lot. And like I said before, whether it's 50% protein expression or 150% protein expression, it's hard to say for any particular gene, but generally speaking, those two cases can be equally problematic. So monosomy, trisomy, both things that ought to be avoided if possible. Now, if we look at sex chromosome nondisjunction, so what we just talked about, Down syndrome being an example of an autosomal nondisjunction in which you have one too few or one too many of chromosomes 1 through 22, you can similarly have nondisjunction of the sex chromosomes, the X chromosome and the Y chromosome. So having extra copies of sex chromosomes, as we're going to see here, is a lot more easily tolerable than having too many or too few autosomes. There's a lot fewer problems associated with it. So this is going to occur with nondisjunction during either oogenesis or spermatogenesis, just like we've seen before. So one particular case of this is called Turner syndrome. The genotype for the sex chromosome is called XO to indicate that there is only one X chromosome, as you can see from this karyotype over here. Now, there are some kind of physical morphological differences that you can kind of see here. This will produce a female. So in terms of wondering, well, is a person with Turner syndrome, are they going to be male or female? Well, they are going to be female for reasons that we'll talk about here in just a minute. But there are some physical, I'm not going to call them abnormalities, but just some kind of distinctions. So females with Turner syndrome tend to be a little bit shorter. They tend to be a little bit more broad in the chest and kind of have some differing morphologies of kind of how their nipples are spaced apart from each other. But otherwise, individuals with Turner syndrome develop and behave very normally. They're of normal intelligence, they have normal function, and usually they have to receive a little bit of hormonal therapy, but otherwise they're able to live very normal lives. Okay, so on the other hand, let's consider a case of sex chromosome trisomy. This is called Klinefelter syndrome, and it's a case of having 2 copies of the X chromosome and 1 copy of the Y chromosome. Now, again, you might be wondering, okay, is a person with Klinefelter syndrome, are they going to be female or are they going to be male? You might be thinking female because they've got 2 copies of the X chromosome, and usually that's what makes you female. And then on the other hand, you might see that they have a Y chromosome, and you might think, well, that's what makes you male. Well, the latter is what's actually true. Having the Y chromosome is what makes you male. Not to get into the weeds of developmental biology, but the default position for human development is to develop into a female. The only way to switch off of that default and instead pivot to developing into a male is to have a particular gene which is located on the Y chromosome. This gene is called SRY, and it encodes a protein that acts as the testis determining factor, and when this protein becomes expressed early in development, it plays an important role in switching off of that female default and signifying male genital development and the development of all of the other male sexual characteristics. So the idea here is that in the case of Klinefelter syndrome, no matter how many X chromosomes you have, so whether it's 2 of them or 3 of them or 4 of them, if you have the Y chromosome and that Y chromosome has the SRY gene intact on it, you are going to develop into a male. Now, Klinefelter syndrome, very similarly, produces some near-normal intelligence and near-normal physical development, but the more nondisjunction you get, the more X chromosomes you start stacking on, the more problems that person would actually run into. Okay, and then finally for this chapter, let's talk a little bit about some different changes in chromosome structure that we can see. Now, these actually have nothing to do with nondisjunction necessarily, so these are actually large-scale mutations in the chromosome. Now, at the end of chapter 14, I think it was, we talked about point mutations, which are single nucleotide changes within the DNA. So it would be like writing a sentence and then scratching out one letter and replacing it with a different letter. These are much, much, much more wholesale changes. This would be like writing an entire book and then ripping out an entire chapter and then maybe moving it to a different part of the book. Much, much, much more significant, much more wholesale changes. So these changes in chromosome structure can be caused by all sorts of things, a lot of them being environmental agents like radiation from the sun or radiation from other places, exposure to chemicals. Viruses can cause chromosome structure changes. So basically, exposure to the environment like this can cause damage to your DNA. And we talked about DNA repair in chapter 14. So if your repair machinery is not able to handle the large-scale chromosome changes that you're seeing, then you end up with a very significant mutation event. So some of the large-scale chromosome structure changes that we'll see here include deletions, in which one or both ends of a chromosome literally get torn off and break off and they don't become rejoined. You can have a duplication event in which one part of a chromosome gets duplicated, and then that causes there to be more than one copy of a particular gene on a particular chromosome, which isn't always a bad thing. Sometimes that's actually an important part of the evolutionary process, as we'll talk about in a later chapter. And then translocation, you have a segment from one chromosome actually gets broken off, and then it moves and reattaches to a different non-homologous chromosome. And usually that's going to follow breakage of two non-homologous chromosomes. Basically, the way the translocation works is that you have breaking off of two different chromosomes, and then the DNA repair machinery doesn't do the proper job of putting them back where they're supposed to go. And then finally, you have an inversion event in which you break off a piece of a chromosome, and then when you reattach it, you actually reattach it upside down so that the genes now occur in the incorrect order, which doesn't seem like a big deal, but the order of genes oftentimes is a very important part of how gene regulation and gene transcription actually occurs, so that can cause some problems as well. So you can see some pictures here that illustrate some of the major chromosome structure changes that we can see. We talked about all four of these: deletion, duplication, inversion, and translocation. Now, to make sure that we understand, these are very different than the much more small-scale point mutations that we talked about before that really only affect one gene at a time. These large-scale chromosome abnormalities should more than likely affect many, many genes at the same time. So in a lot of these cases, especially with deletion, inversion, and translocation, those are going to cause big, big, big problems for multiple genes. So as I said before, genes really need to be in their typical locus in order to be able to regulate it at the transcription and translation level in the proper way. So any of these can be a big, big, big problem. If you want an example of a deletion syndrome, let's look at Williams syndrome, which occurs when a segment is lost off of the end of chromosome number 7. And the major gene that gets lost here that is very important for the etiology of this disease is the gene for a protein called elastin. Elastin is a protein that is found in the extracellular matrix of a lot of tissues, and it gives tissues a lot of elasticity. So when you think about human organs that need to be able to kind of stretch out quite a lot, these affected children that you see here are going to have problems with a lot of those organs. So they're going to have neurological developmental delays, and usually the telltale sign is that they're going to have cardiovascular problems because the tissue of the heart needs to be able to stretch out properly because it is constantly pumping blood. But without this elastin protein, the tissue is not at all elastic, and they have major, major problems with pumping blood. Another one that you can see here called Ehlers-Danlos syndrome, this is caused by a translocation defect. Even though it says inversion syndrome up here, you can just kind of ignore that. That's a typo. This is a translocation defect in which DNA segments from chromosomes 2 and 20 are swapped with each other. So this causes major liver defects, and similar to what we saw with Williams syndrome, it also leads to some major heart problems. So if you look at this picture here, you can actually see what we call nail clubbing, which is oftentimes a very important symptom that indicates improper blood flow and a lot of digestion problems. So you can kind of see how that would cause a problem as well, not having the genes that are responsible for regulating those sorts of processes on the correct chromosomes. Okay, so that's going to do it for Chapter 13. I enjoyed talking about it with you. Join us next time, and we will move on. I think where we're going next in this segment is we're going on to Chapter 18. Now that we've talked about DNA, heredity, Mendel's experiments, and everything else, we're going to start talking about evolution, and we're going to start talking about the nature of DNA and how it leads to changes over time and changes in species over time. So thanks again. Join us next time, and I will see you later.